Hello, I'm Rodney Bennett. Thank you for joining me for today's Nebraska lecture. Since 2003, this distinguished lecture series has elevated some of the University of Nebraska-Lincoln's most notable scholars, researchers, artists, and thinkers. Faculty who live our university's land-grant mission every day and make a positive impact on our communities through their work. Faculty who are committed to making Nebraska, our nation, and our world a better place to live. Our lecturers are experts in their fields, devoted to mentoring and shaping future generations, as well as solving the most pressing challenges our society faces. They embody the spirit of the University of Nebraska-Lincoln and the state of Nebraska. And I am pleased to be able to share this distinguished lecture series with the broader Lincoln community and beyond. It is truly a celebration of our teaching, research, and engagement mission. I am so proud of our faculty's accomplishments and their dedication. Thank you to the Office of Research and Innovation, the university's Research Council, and the Osher Lifelong Learning Institute for partnering to sponsor this lecture series. I hope each of you enjoy today's lecture.
So good afternoon. I'm Nathan Meyer, Associate Vice Chancellor for Research here at the University of Nebraska-Lincoln. And on behalf of the Office of Research and Innovation, it is my pleasure to welcome you to the Fall 2025 Nebraska Lecture. Today's lecture features Dr. Janos Zempleni, Willa Cather Professor of Molecular Nutrition in the Department of Nutrition and Health Sciences. Janos has pioneered studies of extracellular vesicles and their RNA cargoes in milk, and their biological activities within species boundaries, across species boundaries, and across kingdom boundaries, from animals to bacteria. His work shows great promise in improving infant nutrition and delivering therapeutic cargo to pathological, hard-to-reach tissues. Janos received his bachelor's degree and PhD at the University of Gießen in Germany, and he later did postdoctoral work at the Innsbruck School of Medicine in Austria, the Emory University School of Medicine in Atlanta, Arkansas Children's Hospital, and the University of Arkansas for Medical Sciences in Little Rock. Janos joined the UNL faculty in 2001, and he currently directs the Nebraska Center for the Prevention of Obesity Diseases. In 2024, NPOD received a third round of funding from the Centers of Biomedical Research Excellence program at the National Institutes of Health. And this is really a testament to the center's expertise, reputation, and productivity. And as point of fact, over the last decade, center members have secured $210 million in external funding to back that up. During the same time, the number of center-affiliated researchers has increased from 12 faculty in a dozen departments at UNL and the University of Nebraska Medical Center to 53 faculty in 25 departments across UNL, UNMC, and the University of Nebraska-Omaha. Janos also leads the Husker research team that has been very competitive in the NIH's gene editing competition for innovations that could lead to new treatments for both common and rare diseases. And the team recently received the news that they successfully completed phase 3A of this exciting competition. And in his abundant spare time, Janos co-founded and co-owns Minovaca, a biotechnology company that aims to develop technology for targeted drug delivery. Minovaca is working with New Tech Ventures to license this technology. Janos is a fellow of the American Association for the Advancement of Science, and he has received many honorific awards throughout his career. Earlier this year, he was named a Distinguished Fellow of the American Society for Nutrition, and this is the highest honor bestowed by that organization to recognize individuals for their significant contributions and outstanding achievements in the field of nutrition. So Janos, congrats on that. Today's lecture comes at the midst of a very busy month for the Office of Research and Innovation. So just to give you a preview before the lecture begins, our annual research report will be posted online early next week and then available in an abridged print version soon thereafter. And as a spoiler alert, Janos is featured prominently in this publication. Also, be on the lookout for the Research and Creative Activity Report, which many of you in the room have probably contributed to. This also will soon be posted on our website, research.unl.edu. And finally, next week is the annual Nebraska Research Days, our celebration of UNL faculty research, scholarship, and creative endeavors. I encourage you to view the complete schedule online and register for any events in which you are interested in. So thank you for joining us. Thank you to our partners in bringing these lectures to the Lincoln community, to the campus community, the Office of the Chancellor, the Research Council, in collaboration with the Osher Lifelong Learning Institute. So please stay with us after the lecture for question and answer, and then a reception to follow. So please help me welcome our Nebraska lecture speaker, Janos Zempleni. Thank you.
Thanks, Nathan. Well, good afternoon. Most important things first, I have to disclose, start every my presentation with a disclosure. Nathan already alluded at this. I'm the co-owner and co-founder with my partner, Shiantao Guo in chemistry, of Minovaca, which is a company developing exosomes, milk exosomes, for treating or delivering gene therapy to pathological tissues. I have to disclose this, and I have to point to Sarah Quinn, the director of our research compliance office, because she's always breathing down my neck saying, well, you need to make sure to disclose your potential conflict of interest. So I've done this. I wish, or I hope Sarah is listening in to live stream and sees that I'm living up to her expectations. There's a few other disclosures that I have to make. None of them are really relevant for this presentation. It's about grant support, editorial boards, and and so on, advisory services. So I will talk a lot about we are doing, I have done, but the matter of the fact is, this is the group of people who did all the work that I'm going to talk about today. Some really great students and postdocs on our team. Some of them are here in the audience. I saw them. Thank you for coming. The first take-home message of today's presentation is all of them are smiling. So that means it cannot be so bad working with me. Then I also would like to acknowledge some of the funding agencies, USDA NIFA, the Foundation for Prader-Willi Research, the Research SynGap Fund, and NIH, and the one that Nathan already mentioned, NIH Somatic Cell Genome Editing Program, which is really more research prize than research funding. So in our lab, we engage in two lines or two streams of research, both relate to milk exosomes. One of the streams is the importance of milk exosomes in infant nutrition and infant development. The other stream is the use of chemically modified milk exosomes for delivering therapeutic cargo and payload to pathological tissues. By design, I put nutrition to the right side because this stream is a little more murky, and as you may know, nutrition science is a little murkier than the pharmaceutical sciences when it comes down to making decisions and drawing conclusions. So I will not talk about the nutrition area of our research, but I want to show some of the highlights that came out of our lab and some other labs over the years, or the past 10 plus years, showing that depletion of milk exosomes elicits adverse phenotypes, behavior basically, in mouse models and in pig models, and that supplementation with milk exosomes has positive effects on the several phenotypes. And the magnitude of these phenotypes is large. So for example, if you deplete the diet of juvenile mice of milk exosomes, there's a 9-fold decrease in learning and cognition. If you supplement the diet with milk exosomes, the severity of necrotizing enterocolitis decreases by about 50%. That's very important in infant nutrition and premature infants. So today, again, I'm going to focus on the pharmaceutical aspect of our research. The one achievement that my group is very proud of and should be very proud of is that we were selected for a research prize by the NIH's Institute of Somatic Cell Program, Somatic Cell Genome Editing Program, which really put Nebraska or UNL on the map. The reason why I'm so happy about this is we basically competed or outcompeted some of the most prestigious institutions in the country. There was lots of competitors from the University of Pennsylvania, Harvard, and so on. So we really, UNL can keep up with these institutions. That's what I'm trying to say. So I don't expect that many of you have a solid understanding of what what exosomes overall are. So I want to walk you through the biogenesis of milk exosomes or exosomes before I dive into the details of our research. So the way exosomes, and that's not specific for milk exosomes, this is any exosome, originate is where you have two cells, exosome donor cell, exosome recipient cell. The biogenesis starts with an endocytotic event, so basically invagination of the donor cell, taking up something from the extracellular space, and this something then matures into an early endosome, onto late endosome, and these late endosomes end up in what's called MVB, multivesicular body, and in this MVB they're loaded with cargo, and these cargo that end up in the exosomes, in these nascent exosomes, can be RNA, can be lipids, can be proteins, can also be DNA. DNA is more typically seen in exosomes originating from cancer cells, and more often than not is on the outside of these exosomes, not inside the exosome. What happens after loading or sorting is complete, cargo sorting and loading, many of these exosomes are degraded in a lysosome, so they don't even make it anywhere. Some of them are released in the cell cytoplasm or cell cytosol and then change the behavior of the donor cell. And some of them are released in the extracellular space, and that happens when the MVB fuses with the plasma membrane, basically opens up and releases all these exosomes in the extracellular space. Then they travel through the extracellular space to the recipient cell, where they bind to receptors on the surface or fuse with the plasma membrane of the recipient cell, basically end up releasing their cargo into the intracellular space, and the cargo then regulates all kinds of events in the recipient cell, or they're being endocytosed, just like what you had seen in the donor cell, and after endocytosis release their cargo into the recipient cell, which then changes its behavior. So that's for endogenous exosomes. Where we came into this field, that was 2014, when a former student of mine, Scott Bayer from Wayne, Nebraska, he's an NHS graduate, developed an interest in microRNA cargo in these exosomes. That was back in 2014. So that's when we started our journey in the exosome space. And the work that started with Scott and was continued by many, many other folks in the lab showed that milk or that exosomes do not originate exclusively in endogenous synthesis, but can also be absorbed from milk. So dietary exosomes are bioavailable and can actually make it to recipient cells and change the behavior of recipient cells. So, long time ago, but not forgotten. So the title of my presentation kind of already hinted at these exosomes are very, very small. They're about 100 nanometers in size or in diameter. And a question that came up, some theoretical consideration early on in exosome research was, how can these small particles have traumatic effects on a recipient cell? And what I was trying to remind you of is the fact how a nasty little virus, COVID, changed all of our lives. So these coronaviruses have the same size like milk exosomes. And as you all remember, I'm sure, COVID had a huge impact on the way we lived our lives. So, and I think this also in some ways applies to these milk exosomes. As we engaged in the nutritional aspects of milk exosomes, we discovered that milk exosomes possess many, many qualities that make them ideal candidates for delivering therapeutic payload to pathological cells. For example, we showed that these milk exosomes are orally bioavailable, so they can be absorbed from medicine or from the diet. And I'll show you, well, I'm not going to belabor. So these are the publications that support my claims. So what I'm saying here is there's data that have been published in peer-reviewed journals supporting the statement that 50% of milk exosomes are absorbed from the diet or from medicine. And that's an important observation because it enables a very non-invasive delivery of therapeutic cargo. So as you may remember, I'm sure you all have seen these images when cancer patients are being infused with chemotherapy. It's a sad picture because it's an IV infusion. They live in these cancer units for hours. And with our technology, you can actually deliver medicine by mouth. So these exosomes then also cross barriers, not just the intestinal mucosa, but they can also cross barriers like the brain, the blood-brain barrier. So they get it into the brain, and they can cross the placenta, end up in the fetus. So you can deliver therapeutic payloads to the fetus if there is a disease in the womb that requires treatment. Milk exosomes are very, very stable in the gastrointestinal tract, and they protect that therapeutic cargo against degradation. As many of you know, there's lots of pH changes in the GI tract, various acid in the stomach. There's lots of enzymes in the mouth, in the stomach, in the intestine that all are designed to actually break down what enters the GI tract. But these milk exosomes resist degradation and they protect whatever you put into them until they're absorbed. And that's the data, the publications that go with that. They can be easily produced at large scale or at a scale sufficient for delivering medicine to humans. That's always a challenge when you work with cell culture systems, but for our milk exosomes, it's not a problem. One cow secretes, produces more than 20,000 pounds of milk per year, and we can isolate about 10 to the power of 21 exosomes from this annual production. That's one with 22 zeros, and it's definitely sufficient to treat patients. So the scale is not an issue when we work with milk exosomes as drug delivery vehicle. And then, finally, they're biologically safe, which relates to no immune response when you administer these milk exosomes, no effect on liver and kidney function, and no effect on the biosynthesis of red blood cells. So that's an important consideration when you start talking to the FDA and want to get your therapeutics approved for use in clinical trials. Okay, so all these great qualities we see in our milk exosomes, but they do share a few limitations with conventional vehicles used in nanomedicine. And these limitations include that milk exosomes are rapidly eliminated by macrophages, immune cells, because these immune cells recognize milk exosomes as foreign particles, and they start wondering, well, why would they be here? And they start gobbling them up like crazy, which is not great when you want to deliver medicine. And the second limitation that I share with conventional drug delivery vehicles is tissue homing. So that's the question, well, how do we get these vehicles specifically to the intended target site and not end up in destinations or in places where they don't belong and where they cause off-target effects. So again, cancer being a good example. If you're being treated for cancer, you're trying to kill rapidly dividing cells, and that's where many cancer patients actually lose their hair, immune function goes down, having diarrhea. So that's the issue with off-target effects. So let me tell you how we overcome these limitations in our designer milk exosomes. If you look at this is a naive milk exosome. That's how any exosome, that's how an exosome would look like. We call them ME, milk exosome. There's transmembrane proteins like CD9, CD81, CD63 that cut through the membrane 4 times, hence tetraspanins. There's a number of other glycoproteins embedded in the membrane of the milk exosome. So this is our input material, so milk exosomes that we purify from milk actually by using high-speed centrifugation for pre-clearing. That removes cell debris and cells that are present in milk. Then we follow up with additional purification steps, and I come back to them later to get a very homogeneous preparation of naive milk exosomes. And then we convert or functionalize our naive milk exosomes to DMEs. And this is a two-step process. We load these exosomes with gene therapy, so it could be gene editors, CRISPR-Cas, or plasmids encoding genes, and deliver these cargoes to our intended target sites, target cells, in other words. And the second step of functionalization would be the decoration of our milk exosomes with synthetic peptides that enable the targeting to certain tissues or cell types, and peptides that prevent uptake of milk exosomes by macrophages. And these peptides are called don't eat me peptides. That's not a name I made up. I mean, this is just typically if you go to these exosome conferences. So in other words, yeah, okay, so we attach these synthetic peptides by forming a hydrazone bond, and we did report or did show and demonstrate that this hydrazone bond peptide to exosome is stable. So it's not degraded in the GI tract, it's not degraded in plasma, and it's not degraded in the stomach. Okay. So I have one slide for you on the don't eat me story. So if you look at our milk exosome, so this is the milk exosome loaded with medicine. And you see this macrophage right here trying to eat up the milk exosome. And these macrophages are super efficient when it comes down to eliminating milk exosomes. That was demonstrated by another former student. She's currently a postdoc with the NIH, Afsana Khanam. She reported, or she identified the transporter that actually facilitates the uptake of our milk exosomes, and demonstrated that this transport is extremely efficient. There was no way we could even saturate the transporter, so it kept on taking up the exosomes and eliminating the exosomes from culture media or from a mouse model, or in a mouse model. And but we also devised the technology, or we utilized the technology that enabled or prevented the uptake. So for those of you who have seen Harry Potter, you probably remember the invisibility cloak, and that's kind of the analogy that we're using. So we actually hide these exosomes, DMEs, by covering them with an invisibility cloak, which is a synthetic peptide in our case, CD47. And that worked like a charm. Another former student, Alice, and she's currently with UNMC, working as a research scientist. This one is a little complicated to explain. It's basically the plasma levels of fluorophore-labeled milk exosomes. Orange are the milk exosomes that are masked with a cloak. So they are very high in plasma, so they're not eliminated basically. These are the exosomes not decorated with the don't eat me peptide, and their plasma levels tend to be much, much lower than the CD47 positive exosomes. So in other words, we can definitely prevent elimination of these exosomes from plasma. The next panel is probably a little more intuitive. What you see here is the brain of different mice, not these mice, but different mice, either being treated with CD47 positive milk exosomes, or the negative control, which is CD47 negative exosomes, and the solvent. So solvent, basically no signal. The CD47 negative exosome, there's a little bit of uptake, bright red, into the brain. And you can see if we decrease macrophage elimination of our exosomes by CD47 decoration, the uptake in the brain is way, way higher than what you see in the CD47 negative, and the penetration of the entire brain is great. So it gets everywhere in the brain. So the first, don't eat me. So now talk about the homing. So you would have to imagine, I think, the tissues that you want to target as, having zip codes. So each tissue, like the heart, has a zip code, the lungs, the liver, the stomach, and so on, brain. And what we do in our milk exosome technology we use the milk exosomes that you see here, slap a shipping label on them, which is a homing peptide, and they know and understand to which address they need to be delivered and where they should not go to. I think this is a slide that tells it all about homing. So this is again work done by Alice in collaboration with two folks at UT Southwestern, Bob Baku and Denise Ramirez. What we did in this study was Bob actually created a mouse xenograft model of human glioblastoma. If you know cancer, this is the most deadly form of cancer. Stage 4, you have a few months to live. And they stained the tumors. So basically they took the tumors from a human patient, injected the tumor cells into the mouse, and here you see the tumor growing in the mouse brain. And and the same mouse then got our milk exosomes stained with a different dye. And you see here is where the milk exosomes are, and I'm sure you can already imagine what I'm going to say next. If you merge these two images, the exosomes, I should have mentioned this, they were decorated with glioma homing peptides, COOP or ApoE. So the next step would be, okay, merge these two images, and you can see the yellow is the co-localization of milk exosomes and tumor, and you can see co-localization is almost perfect. So all these milk exosomes go to the tumor, very, very little, if any, in the surrounding healthy cells. And Bob was telling us he has never seen anything as specific as what we have done with our milk exosomes. So brain cancer is an interesting field, an important field, but we also developed an interest very quickly into the treatment of rare diseases. And that interest was enabled by a good friend of mine with the SynGAP Research Fund. He reached out to us saying, okay, I've heard about your milk exosome technology. Is this something that you could use to deliver gene therapy to our children, basically? And so I was really thankful they recognized the merits of our technology and started supporting our research and still supporting our research. And that opened the door for us to work with other rare disease patient advocacy groups, currently supported by the Prader-Willi syndrome and Scharf-Young syndrome foundation. We are in the final stages, hopefully, of striking a deal with the Cure San Filippo patient advocacy group. And we submitted a letter of intent and wait for a response to the Alpha One Foundation. So these are all rare disease patient advocacy groups looking for treatments and cures for their patients and for their children. So rare diseases, as you can imagine, are rare. It's kind of it's obvious, and So, but only if they are considered individually. So the definition of rare diseases varies by region. Here in the US, a disease is considered rare if it affects fewer than 200,000 people. In Europe, it's different. It's a certain number per 100,000, and I don't know what the number is, but basically here in the US, it's fewer than 200,000 people in the US living or suffering from a particular disease. They're underserved by the pharmaceutical industry. Well, I did consult, or had confirmed my assumption by AI. And of course, they're underserved by the pharmaceutical industry because the high cost of drug delivery is difficult to recoup from a small number of patients. If you have only 500 patients, like with SynGAP research deficiency or haploinsufficiency, the pharmaceutical industry will never get its money back. Because it costs hundreds of millions of getting these treatments through clinical trials. Taken together, though, all rare diseases together, there's about 25 to 30 million Americans suffering from 7,000 rare diseases. And these are the patients, not counting the caregivers. So imagine for each child suffering from one of these diseases, there's at least one caregiver in the family, typically, who gave up on his or her career. And they stay home. These kids are very, very difficult to deal with. They require full-time care. And that's why our DME technology is so beautiful. So this you have seen before. This is how the DME looks like with the homing peptide and the therapeutic cargo. And the versatility of our system is, if you see, we can change the cargo any way we wish. So this is from pink to red to yellow to orange. And so in other words, we can load whatever we want, whichever gene we want to load. And then we can also modify our homing peptide. We can send these DMEs to any tissue that we want them to send to. So this is illustrated by using orange, green, black, brown. So one example from the lab, really something that Camar in the audience did. So this is about the SYNGAP research, SYNGAP haploinsufficiency. This is a disease where one allele of a gene is mutated. Typically, as many of you know, you have two copies, in most cases, you have two copies of a gene per cell in most tissues. It's not always the case, but for the most part, it's the case. And in these children, in a mouse model here, only one of these copies is functional. So what you see here is the protein band. If you run samples on the gel and you probe them with an antibody, you can see, okay, this is SYNGAP protein in normal mice, or wild-type mice. And we did not treat them here. So this is an untreated mouse. This is a mouse sample brain that was given empty milk exosomes, so no plasmid. Then you have this SYNGAP haploinsufficient mice, also brain samples, and you can see there's a faint signal, protein signal, about half of what you see here in the wild-type mice, which is to be expected because one copy of the gene does not work and the other copy still makes protein. So it's down to about 50% of what you see here. And this is again untreated mouse brain. This is a brain from a mouse that was treated with empty milk exosome, no plasmid. And this is a mouse brain, a brain from a mouse that was treated with a plasmid encoding SYNGAP1. And you can see, a crazy amount of protein was produced, way more than what you see in the wild-type mice. And what you want to be, or where you want to be, is really what's boxed here in yellow. So this is the point where you want to get, and we, of course, way overshot. It was a good problem to have. You don't want to go too high because these things mess with brain chemistry, so they could have adverse reactions, effects if you go too high. So Camar went through 3 rounds of titration. First round, she brought it down a little closer, blue versus yellow, but it still was about 3-fold higher than we wanted to be. And then the second or third round of titration actually got us to where we wanted to be. So basically a ratio of 1 on average, here blue versus yellow. So in other words, you can really titrate the dosage that you want to give to mice and hopefully at some point also children. Talk a little bit about homing, or hypothalamus now. So this is a region of the brain that plays an important role in Prader-Willi syndrome, another rare disease that we are trying to treat. So what we did here is, what you see here is tissues from mice that we harvested, and these are tissues from mouse that were treated with solvent, basically saline, no signal, basically. You can see, very faint, so blue means weak signal. And liver typically produces some autofluorescence, that's why it's green. And that's not so important, this is not so exciting, this is just the solvent control. The middle column is tissues from mice that were treated with milk exosomes without a homing peptide. So they go anywhere where naive milk exosomes would go. You can see there's a little bit in the brain, there is a lot in the lung, a lot in the kidneys, spleen, and liver. And then the right column that you see right here is milk exosomes decorated with a homing peptide that is supposed to take them to the hypothalamus. And you can see enrichment in the brain increases considerably compared to the undecorated milk exosomes. And what's equally important is that the enrichment of exosomes in off-target tissues, in this case lung, decreases basically to background level. So you minimize off-target effects by decorating these milk exosomes with a hypothalamus homing peptide. And the same is the case, I think, for the liver. I think I boxed that too. Okay. Liver is a major sink for milk exosomes. because of the immune cells, the Kupfer cells in milk, in livers. And you can see it goes from very strong signal, lots of red, to very, very little signal in the homing peptide decorated milk exosomes. More granular presentation, different regions of the brain. So basically dissecting this brain, the sample, looking at different regions, cortex, cerebellum, hypothalamus. What you can see here is the hypothalamus with the homing peptide is much stronger than the hypothalamus without the homing peptide, and not much else is going on in these other regions of the brain. So now we wanted to deliver the gene encoding for one of the proteins deficient in Prader-Willi syndrome to a mouse model of Prader-Willi. And we started by cheating a little bit. So we thought, okay, let's try the delivery by nasal spray. And it was well intended. I mean, we thought, okay, nasal spray is very non-invasive. It's really just a puff of a spray with milk exosomes to deliver the gene. You don't bother anyone. And the benefit also would be nasal delivery. These exosomes have to just cross this very thin layer of cells, the olfactory bulb, to get right into the brain. So it would be very, very effective, we assumed, and that proved true actually. But then we were told by one of my friends from the SYNGAP community, well, this is not going to work for my son. I mean, these kids are very aggressive. You would have to wrestle your son in order to get the spray administered. And he says, oral is the way to go for us, because then I can just dump it into his drink and he will drink whatever is I put in front of him. So nasal he didn't like so much, and we had the data already for nasal spray. So this is Prader-Willi syndrome. We administered a plasmid encoding MAGEL2, which is the protein that we're talking about, to a mouse model of Prader-Willi. And what you can see here, it goes to the brain as expected, and the red signals, this protein is expressed very effectively in the brain. These are the control mice. This is one mouse which was given empty milk exosomes. It's just autofluorescence that you see here. The disadvantage here is not just to fight your kids for delivering or administering this milk exosome. It's also this clustered expression, so right around this olfactory bulb that you see here. So it doesn't really penetrate the entire brain. So after being told about oral is the way to go, we attempted oral delivery. So we delivered the same gene using the same exosomes, the oral route. And what you can see here, again, empty milk exosomes, no plasmid, the brain. These are the plasmid-loaded milk exosomes given to the mouse by oral gavage. You can see, okay, this exosome delivered MAGEL2 fusion protein to the brain, and actually is successfully expressed in the brain. That was really, very exciting for us. Then we tried, this was a single dose, and we tried, okay, does multiple dosing increase expression? And I think if you compare this one, multiple dose of milk exosomes, to the single dose, multiple dose does not really make much of a difference. The signal is pretty much the same, which means, it really doesn't benefit the mouse or the future patient to give them multiple doses of the milk exosome. We assume that's because neurons in the brain don't divide very much, if at all. And once you have the plasmid in the brain, it will stay there and just keep producing protein. So we did quantify the expression. Now this time is DMEs with a hypothalamus homing signal delivered to this mouse. And you can see, if there's no DMEs given to the mouse, this is the expression of MAGEL2 that you have. And if you give the mouse or another mouse the milk exosomes with a hypothalamus homing peptide, dissect the hypothalamus and measure the MAGEL2 expression, it's about 92% higher compared to the control mouse, which is, pretty impressive, I think. And here I'm going to show the Western blot that goes with the data. The reason I'm doing this is the Prader-Willi community has been struggling for many, many years to develop an antibody to detect MAGEL2. And so they paid one of these leading antibody companies a lot to make the antibody. They couldn't do it, but in our lab it worked. So we actually generate an antibody that will benefit the Prader-Willi community for years to come, and will donate the antibody or the plasmids encoding the antibody to some repository to make it available to the entire Prader-Willi community. So it's not just the brain that we can deliver our cargoes to. We can also, and I'll give you a couple of examples, deliver it to the pancreas and the heart. What you see here, same layout as before. This is really just the vehicle control, basically saline treated mice, various tissues. And here you have the milk exosomes not decorated with any homing peptide, going to various tissues. And here you have the milk exosomes decorated with a pancreas homing peptide. And pancreas, I boxed it for you, normally is a really poor target for naive milk exosomes. There's very little delivery to the pancreas. But if we decorate our milk exosomes with a pancreas homing peptide, lots of milk exosomes actually go to the pancreas. And again, negative control, or off-target control, the liver, very, very effective sink for milk exosomes, which is not what you want. And if we decorate our milk exosomes with a pancreas homing peptide, very little actually ends up in the liver. Exactly what we want. I don't want to belabor this too much. I mean, same story basically with a heart homing peptide. You can see the increase of milk exosomes in the heart without decoration compared to with decoration, and the disappearance in an typical off-target tissue, the spleen. So very strong signal if not decorated with a heart homing peptide. Very, very weak signal in the spleen if decorated with a heart homing peptide. Okay, so far I talked about the delivery of genes. This research prize that we got from the NIH Somatic Cell Genome Editing Program is not really all that much interested in gene delivery. They want to deliver gene editors. So in other words, what we had to do is demonstrate we can deliver CRISPR-Cas to tissues. And I think it's great to be part of this group. It's supported by NIH Director Common Fund, which means this is high priority. It's cutting-edge biomedical research that is really at the forefront of what NIH wants scientists to do. Anyway, and that coincides with a very recent move by the Food and Drug Administration, where they said, okay, they want to speed up the approval of gene editing therapies compared to early days, where there was really a complicated approval process. For those of you who are not biomedical scientists, gene editing means you know of certain mutations in a patient's genome, and you take tweezers to very specifically remove the dysfunct genes and replace them with functional genes or sequences of genes. So basically cutting out what's not supposed to be there and replacing it with something that should be there. Okay, there's two types of gene editing that you could engage in or could test your technology with. There's loss of function versus gain of function. Loss of function is basically turning off a functional gene, basically extinguishing the fire. Turning on a gene is starting a fire. So in other words, loss of function versus gain of function. What we ended up doing, and that was actually Barbie, also a student in the lab, currently in the lab, was loss of function. So we started with cell culture systems, and I'm not going to belabor. This was challenging enough, but I'm not going to belabor this. I'll move right on to the mouse model. Barbie, sorry, I know you like another slide better than this one, but this is my favorite of the ones that you produced. So what you see here is basically we decorate our milk exosomes with a brain homing peptide, loaded them with CRISPR-Cas that does target a fluorescent protein called EGFP. So these proteins have a bright green fluorescence, and when you deliver the gene editor, loss of function, remember, so the light in the brain is turned off, and the light in all these off-target tissues stays on. So very, very specific delivery. Same with the heart. So Barbie decorated the exosomes with a heart homing peptide, turned off the light in the heart, and the light in all these control tissues stayed on, which is perfect. And then the pancreas homing peptide, she dimmed the light in the pancreas, not quite as dramatic as heart and brain, but again, controls very, very bright. The reason why I think this was not as effective, or doesn't seem to be as effective as heart and brain, is grant submission deadline. So we had, I think it was 9 days, if I believe, after treatment. Here was 9 days, and here was about 6 days. So it's probably just a matter of half-life of the protein, but it works, so the light goes down. So we always have to compare our technology to conventional gene therapy delivery protocols or technologies. And so when you compare stability in the GI tract, including oral bioavailability, DMEs check out. Lipid nanoparticles, they're not stable in the GI tract, and they're not orally bioavailable. Adeno-associated virus, also not stable in the GI tract, and also not orally bioavailable. So these two need to be injected. Safety. So DMEs, to date, there's no evidence of DMEs eliciting an immune response. LNPs elicit severe immune responses. AAVs, you cannot administer multiple times. So patients start to develop antibodies to AAVs once you start treating them. So you cannot do repeat treatments if that's what you need to do or what you want to do. Neutralizing antibodies, so this is basically pre-existing antibodies. So if a patient was never exposed by a physician to treatments, are there any antibodies in the body that would eliminate these delivery vehicles? Not the case for DMEs, not the case for lipid nanoparticles, but for a relatively large fraction of the population in the US, it's about 20%, have been exposed to AAVs, adeno-associated viruses, in their lives. So they have pre-existing antibodies, so they will eliminate the treatment right because they don't like these AAVs in the system. And then finally, payload size. So with the DMEs, we've successfully used plasmids up to 12,000 base pairs, which is great. And then LNPs, the current state of the art is about 7,000 base pairs, and they're pushing to go higher. But I mean, right now it's definitely smaller than DMEs. And the AAVs, it's about 3,000 base pairs, so really cannot load a whole lot in an AAV. So we like our technology for these reasons better than the competing conventional technology. This is just my cheat sheet. If anyone started probing questions after the presentation about these technologies, I would have to go back to the slide and try to remember or remind myself of why these LNPs and AAVs are not as great as our DMVs. Okay, so food and drug administration. In the past, when you approach them about exosomes and treatment of patients, one of the big questions they always asked was, well, I mean, these exosomes are heterogeneous. So if you purify exosomes from some matrix, it's not just exosomes, and that causes a big problem in drug dosage, because you think you give 100% exosomes, but it's really not 100%. And that's a table I pulled from one of my grant applications. So when you work with milk, which is a very difficult matrix to work with, there's exosomes, there's ectosomes, there's lots of other relatively small particles that sometimes co-purify with exosomes, which is the problem that you don't want to have. The way you can relate is probably if you put a human milk on a bench top or a countertop, after a while the cream phase starts to separate from the plasma phase, so which is the skim milk. That means there's lots of lipids in milk, and there's the fat globules. So you really want to separate these exosomes from these other particles in order to minimize the heterogeneity of exosomes. Good example is probably exosomes versus ectosomes. Exosomes are a little smaller, up to 100 nanometers diameter. Ectosomes are about 100 versus 1,000, up to 1,000 nanometers. And they have the same density. So you cannot really separate them by density, which is what you would do with ultracentrifugation or some other centrifugation technologies. But you can separate them by size, which is size exclusion chromatography. I'm going to show you the data about that in a minute. Actually now. So this is something that you've seen before, kind of a cropped part of a slide. So we said, okay, we pre-clear our milk, naive milk exosomes with this high-speed centrifugation. Then we apply two additional technologies, size exclusion chromatography and sucrose density gradient centrifugation. I'm not going to tell you what these are, but basically when you go through this procedure of successive steps of purification, you can see the enrichment of a typical exosome marker protein from very, very little to very high after pre-clearing SEZ and SDGZ. So you really enrich for exosomes. At the same time, you deplete of a typical contaminant, which is a whey protein, beta-lactoglobulin, very high at the beginning in the starting material, and then non-detectable after the final step of purification. And this is two Western plots to illustrate how this looks like in the lab. Then there's also, and I kind of brushed over this at the beginning, there's an optional purification step that we built into our system. You may have caught there is mention of His6, which is 6 histidines that we have built into our homing peptides. And this is how the peptide looks like. So this is the example of a pancreas homing peptide with the amino acid sequence. And this is the His6. And this is the hydroside that actually creates the bond to these proteins. And His6 in molecular biology is known to bind to nickel resin. So you can actually easily purify these labeled exosomes from any matrix by nickel His6 purification binning. So basically you know that everything that you pull down with nickel 2+ has the peptide and goes into the medical bottle. Everything that has no peptide will also not have the His6 and goes into the trash bin. You can sell it online if you want to, if you have no shame. But so basically this is useless. We know these don't have the peptide. The other advantage here is that you know everything that you pull down is not only labeled with a peptide, but it also has the peptide that, in this case, will take the exosome to the pancreas. So in other words, it's very, very specific purification, and you know exactly what you got, and you got what you want, basically. So then there's two questions that popped up, or that came up when we worked with the His6 mark tag right here. One question is, can we really purify these exosomes from our matrix? I have to take a look at my clock. I have some time. And we can. So basically what you see here is, this is the mix of, fluorophore labeled DMEs with the nickel resin, and you can see bright red because it's fluorophore labeled. This is the control, C, and this is not fluorophore labeled. You can see, okay, this is the starting material. Then you wash the nickel column or the nickel resin just with solvent, and you can still see, okay, the DMEs, bright red, and here just a very minor autofluorescence signal for the control. You wash the nickel resin with imidazole, which elutes the supernatant or the His6 containing compound from the nickel resin. And you can see when we harvest the supernatant after the first wash, lots of exosomes in the supernatant and nothing in the control sample. We washed the second time and still got quite a bit of labeled exosomes of the nickel resin and really nothing of the control. And this is the remaining resin, so basically showing no signal, which means we got everything off the nickel resin with our imidazole. So this is a very effective way to purify these DMEs. The second question that came with these His6 containing peptides is, does the His6 somehow impact the ability of the homing peptide to take the exosome to its target? In this case, remember, I said it's a pancreas homing peptide. What you can see here is the pancreas from a mouse that was treated with solvent, so basically no signal. This is the pancreas from a mouse treated with non-decorated, undecorated milk exosomes that were fluorophore labeled, kind of a modest signal, just like we saw before. And this is the pancreas of a mouse treated with a pancreas homing peptide that did not have the His6. And you see lots of enrichment in the pancreas. And for comparison, this is the mouse treated with DMEs decorated with a pancreas homing peptide that did contain the His6. And these two signals are basically the same, which means the His6 did not impair the the functionality of the pancreas homing peptide. So you can still get these exosomes to the pancreas. So much of the work that we have done, or that I've shown so far, is in mice. Then, of course, the question is, well, do you want to treat mice or save mice lives? And I don't know. I mean, we really want to take this to the human. And then some folks ask, well, try to work with species other than a mouse, because mouse is the low-hanging fruit when it comes down to crossing the blood-brain barrier. You should work either with swine or dogs or non-human primates. And you can see here the brains of these 3 species. And again, another table from one of my grant applications comparing features in the human, swine, and mouse brain. And I'm not going to read through these lines here, but basically the message is the pig brain is similar to the human brain. The mouse brain is a good model for the human brain, but it's not nearly as similar as the pig brain is. So in other words, pig is a better model for our studies. We collaborated with a person at UC Davis, Ras Hovey, and sent him our milk exosomes overnight. Milk exosomes that were decorated with a neuron homing peptide. So, neuron homing peptide loaded with a fluorescent protein, or not protein, with a plasmid encoding a fluorescent protein, IRFP. He delivered these exosomes to his piglets age 2 days by syringe feeding, so it's oral administration, harvested the brain and some other tissues. And what you can see here in the first piglet, there's definitely the expression of the IRFP protein, bright red. And here in the second pig, same story. The control pig, which did receive milk exosomes without plasmid, did not express any of these IRFP. So in other words, our technology worked in pig models too. Okay, then we also doing some, what a friend of mine from SynGap tells me, some of the more mundane work. And one of these things is to look at endotoxin levels, because endotoxins, they elicit an acute inflammatory response if given to animals or given to humans, and that's of course not anything that you want to have when you treat your patients. These endotoxins originate when gram-negative bacteria break down, so it's a toxin embedded in the cell wall. So in other words, that's where the endotoxin comes from, bacterial contamination. I tried to look up a picture for you of a mouse responding to endotoxin exposure, but AI informed me, there is no such picture in the public domain. It's too gross. And so we collaborated in order to keep our endotoxin levels low. We collaborated with UNL Food Processing Center. They have this high pressure processing instrument or machine, which is a very gentle way actually to sterilize, or well, decrease bacterial load in any matrix. And we did this in combination with membrane filtration. So this one kills bacteria, but does not eliminate spores. This one will eliminate the spores from samples. So we did both of them in combination. And what we saw was endotoxin levels less than 0.05. And that's work done by Kamar and Barbie. They did it twice, and I think it was the first attempt, which is great. This is well below levels that are accepted by the FDA. The second one was even lower, so I think we can definitely say our preparations are endotoxin low, and safe. And there was, I think, some other thing that came on this slide. Yeah. So then we also wondered, okay, does this high pressure processing decrease the number of exosomes in our samples? Because we don't want to lose our exosomes, we want to keep the exosomes. And you can see this is before HPP, before treatment. This is after treatment. That's work done by Jin Yuan, PhD student at the lab. And so basically we did not lose any of these exosomes by treatment with this high pressure instrument. Jin Yuan also cultured these exosomes in collaboration with Tash on agar plates, and neither gram-positive nor gram-negative bacteria were detectable after the combined use of high pressure and filtration. So basically no microbial contamination. So mundane work continued. it's not terribly difficult to make these exosomes, but it's difficult enough that remote hospitals or underserved areas probably cannot do the trick. So we would have to prepare to ship our samples to remote areas, and these exosomes without proper treatment are not super stable. So we have to find technologies to preserve them during storage and during shipping. It's also something that NIH would, or FDA would want to ask, or would want to see at some point. So if you see these remote hospitals, I mean, it's probably a little bit of an overstatement here, but you have to get our technology to these places. And what we ended up doing, it's not rocket science. People do this in the exosome field all the time. We add trehalose, which is a carbohydrate, FDA approved for use in ice cream because it prevents the formation of ice crystals. And you can see at a level of 3% trehalose in our preparation, we can actually freeze these exosomes without any loss. So this is the fresh milk, and this is 3% exosome. They're about the same. Lower doses of trehalose or no trehalose, yeah, you lose the exosomes. So we used that technology in the study that I already showed you with the swine in California. So we froze the exosomes here in the presence of 3% trehalose, stored them for, I think, a couple of days, shipped them overnight to Davis, California, and you have seen this before. I mean, they stored them probably also for another 1 or 2 days, delivered our exosomes to the piglets, and they delivered functional payload to the brains of mice, so of pigs. So basically, I'm saying is, trehalose preserves not only the numbers of these exosomes, but also functionality of exosomes. So now, not quite so mundane. Another new student in lab, Hui, is looking at genome integration. I mean, so basically asking these plasmids, which are circular pieces of DNA, do pieces or fragments of these plasmids integrate in the host DNA? And that's what's illustrated here. Could be either a part of the gene that you want to deliver, yellow, or could be just another part of the plasmid. And they sometimes land in very weird places in the host genome, and that caused a big problem in the early days of gene therapy, including fatalities. That was in 2001, I think it was in France, because of these fragments of the plasmids landed in places where they should not go, disrupting important genes or turning on cancer genes. And so what we are trying to do, and that's work in progress, is work with non-integrating foamy virus vectors that will not actually have much lower frequency of genome integration into the host genome. Okay, future steps. We're almost there. So we want to expand our swine study, waiting for the go-ahead from California. We are engaging in a discussion of an investor paying for a study in non-human primates, including single-cell RNA sequencing analysis, which is a technology that would enable us to tell exactly in which cell type in a brain our cargo would go. So basically, we'll be able, okay, hypothalamus is really going to the neurons in the hypothalamus as opposed to astrocytes. So that's very expensive, but also very informative. Then we will engage in gain-of-function editing of genes implicated in rare diseases, specifically probably SYNGAP haploinsufficiency. So we have a mouse coming in from a colleague that enables these studies. Then we hope to have an interact meeting with the FDA. So these are the meetings in which the FDA will tell you, okay, this is what we want to see if we want to consider your technology for investigational new drug status, which is then the next step. So after interact, we hope to advance to investigational new drug approval by the FDA, which would be a step toward clinical trials. So once you have IND status, you can engage in small clinical trials, in small numbers of human patients. And that's where the rare disease communities are very, very helpful, because some of these diseases have very, very severe phenotypes. There's no treatments, and so the FDA is always a little more accommodating when it comes to treatment of rare diseases compared to other diseases. If you have deep pockets, call this number. This is my wife's phone number. She's here in the audience. She's answering the calls day and night, 24/7. Reach out to her if you can read this number. 24/7, any time of the day, correct? So I've been in Nebraska for more than 20 years now, since 2001, as Nathan was saying. I quickly realized biomedical research is appreciated, but this is an ag-heavy state. So this is my shout out to the ag community. So the dairy industry will probably also, or stands to benefit from our technology, because we hope at some point in the future to isolate our milk exosomes, the starting material, from a waste product in cheese making. It's the whey fraction, basically the supernatant that you have left behind when you make cottage cheese. And the dairy industry pay lots of money, 6 to 7 million to put up a waste treatment system for getting rid of this waste, and up to 1.9 cents for each of the 300,000 gallons of milk per day. So I didn't need AI to do the math myself here. So 300,000 times 1.9 cents translates into about $5,700 per day in operating expenses for these mid-sized dairy plants, plus the $6 to $7 million upfront for building the waste treatment system. So that's number one. So the cost to the dairy industry would go down because they can actually use this for exosome purification. And then the income would also go up because all of a sudden milk changes from a food to a medicine. So dairy farmers can sell their milk at premium prices. Right now, they're being paid, or 2024, they were paid about $23 per 100 pounds of cow's milk. Compare this to the cost of cancer drugs, it's about $24,000 per month in the United States per patient. So there's definitely a premium price that dairy farmers could collect. So with that, I'm open for answering your questions. Thank you.
Thanks so much, Janos, for that masterclass on milk exosomes and all the important breakthroughs coming out of your lab and in collaborations with others. We probably have time for 1 or 2 questions before we break for the reception. Does anyone have a question that they're yearning to ask Janos? Amy's got one.
Thank you, Janos. That was great. You didn't mention the sex of your mice. Is there any specificity with the homing based on sex?
Well, good question. So we looked at sex in our bioavailability studies and did not see any effect. We did not consider sex as a confounder in our homing study. So we don't know. I mean, bioavailability-wise, no effect of sex. Homing, we just don't know. And we use a mix of sexes. So some of this was done in females, some of it was done in males. And we never really noticed an obvious difference in male versus female.
Christian. You're fighting over who's going to get the mic, Christian.
As a layperson, I have a small cluster of questions. So you showed in the introduction that within a cell, exosomes are sometimes degraded again, and then they are spit out of the cell. And I wonder, what is their natural function? Is it also payload delivery to other cells, or what are they supposed to do?
Yeah, it's a great question. Intercellular communication. So basically, outside the milk realm, let's say there's exosomes traveling from fat tissue, adipose tissue, to the liver, and telling the liver what to do, how to respond to a certain environmental impact. So, for example, there's a lean mouse or a lean human versus an obese human, and then the question is, how does the signaling change? Do the adipose cells, adipocytes, send any message to, for example, to tissues implicated in cancer, obesity-related cancer, basically colon, mammary gland, and then basically asking the question, the question that we are asking is, do they actually turn on the cancer risk or increase the cancer risk? So cell-to-cell communication, basically, that's what I'm trying to say. That's the natural function of these exosomes. And the thing is, I mean, that's where the nutrition part came in. They go to the brain very effectively. And I don't know if you remember what I was saying about the phenotypes. Many of the strongest phenotypes of milk exosome depletion, but also supplementation in nature and in not drug delivery and in the nutrition field really relates to the brain. So these things go to the brain. The brain does not develop very well or gets very sick actually if we take the milk exosomes out of the mix for mouse.
Other questions? Janos, I'm going to take the privilege of asking the final question. And you really set the stage nicely talking about sort of the need for treatments for rare diseases. What sort of motivates you to be working in that space?
Okay, it was a good question. I think initially it was really this one person reaching out to me asking, well, I've heard about your technology. I would really like to see you use it for the good of our children, basically. Then I attended, I think, 3 conferences of rare disease communities over the years, and it's a sobering experience. So they typically have a very different approach to science meetings. There's typically one science day where people come in and talk science, and the next day it's patient or family days. So they bring the parents with their children. You see all these little kids, and you can tell they're not doing well. I mean, so it's really, you see it with your own eyes saying, well, this is something worthwhile doing. Okay.
All right, with that, I think we're going to draw today's Nebraska lecture to a close. Let's thank Janos for being selected as the fall Nebraska lecturer. Once again, thanks.